For the sixth video lecture of Chapter 3, we're going to talk about some of the bacterial internal structures, and please read along in your textbook if you haven't already done so. Most of these we're just going to kind of hit some highlights on, and then I'm going to spend a little bit more time talking about one particular structure called an endospore that does not exist in all bacteria. All right, so first of all, let's talk about the cytoplasm. You've probably heard that term, no doubt, before if you've ever had any kind of biology class. So everything that's internal to the cell membrane there, the cytoplasmic membrane, all of that is the cytoplasm. And sometimes people confuse that term with cytosol. That is referring more specifically to the fluid substance that exists throughout the inside of the cell. So cytoplasm is the cytosol plus all of the other components inside the cell. If you could scoop this out of the cell, it would probably have a jelly-like consistency, because there's so much stuff jam-packed in there, it's going to be kind of thick and jello-like. All right, so what kind of structures do we have inside a cell if we are a prokaryote like bacteria are? So of course we have that fluid, the cytosol. Then there are, so this little yellow sphere here, that's supposed to represent an inclusion body, and we'll talk a little bit about those. Some bacteria have those and some don't. Ribosomes, so all these little specks you see everywhere, little blue specks, those represent ribosomes. Those are basically the little tiny machines inside cells that build proteins. And cytoskeleton, so the diagram that your textbook has developed, all these little kind of pale, thin lines that are scattered around everywhere, and it might be difficult to see that on the video. You need to take a look at it in your textbook or on the actual PowerPoint file. That represents the cytoskeleton that exists in some prokaryotic cells. And then finally, endospores, not shown here on the diagram, but endospores do exist in some bacteria, and we'll talk about those because those are actually very, very clinically significant. Something else I don't have listed here on the PowerPoint slide, this purple string here, that represents the DNA of the bacterial cell. So that's also known as the chromosome of the bacterial cell. That's where all the genes are located, those genetic blueprints. And then over here, there's a little tiny circle over here that looks multicolored. Again, it might be kind of hard to see on a small video, but that represents an extra piece of DNA called a plasmid that exists in some types of bacteria. It looks like I gave away quite a bit of this information already over here on this slide. Anyway, the cytosol, again, is the liquid part of the interior of the bacterial cell. So guess what its main component is going to be? It's going to be water. You know, living things are water-based, at least the ones that we know about here on Earth. Also, dissolved substances, solutes. Remember that term, solute? Anything that's dissolved in a solution. And so we have lots of solutes inside the cell. That includes ions, things like magnesium and potassium and sodium and chloride, things that you learned about before, various acids and bases, waste molecules, carbohydrates, proteins. All of those different things that are the chemical components of a cell are going to be dissolved within the cytosol, and so jam-packed in there, it's going to give it kind of a thick consistency. All right, again, this little purple squiggly thing here, that represents the chromosome. Most bacteria just have one chromosome, one big, large chromosome. It's a DNA molecule, so it has those properties that we've learned about with regard to DNA back in Chapter 1, but it actually exists in a circle, so it doesn't have free ends. The molecule, the ends are actually joined together, and then that circle is all wadded up, like you see there in the diagram, because it's actually really big. If you could take that DNA molecule out and stretch it out, it would be many, 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 many times longer than the bacterial cell itself, so that has to be wadded up and jam-packed inside there. And again, that's going to be where the genes of the bacterial cell are located. And again, that little circle that you see right there represents a plasmid. That's an extra piece of DNA. Some bacteria have those and some don't. When bacteria do have those, they generally have extra genes on them that allow that bacterial cell to have traits that it did not have before. Those traits usually help the bacterial cell with survival. So it could be things like antibiotic drug resistance, the genes that allow a bacterial cell to become resistant to a particular type of antibiotic drug like penicillin. Many times those types of genes exist on these little extra pieces of DNA that are called plasmids. If you guys remember the sex pilus or conjugation pilus, that little tube that some gram-negative bacteria make and extend out and attach to other cells, and we said they could transfer DNA through those. A lot of times it's these little extra pieces of DNA that get transferred through those tubes. And so this cell might be resistant to ampicillin, or it might have a resistance to some sort of antiseptic or disinfectant chemical. And if it transfers that plasmid over to a cell that doesn't have that little piece of DNA, now that other cell may develop those traits as well. So we'll talk more about that when we get to Chapter 8 and we cover genetics. There's some genetic hanky-panky that goes on between some types of bacteria. Not going to spend a lot of time on inclusion. So again, that little yellow sphere there represents kind of a generic inclusion. Some bacteria have those and some don't. Not necessarily that size. Sometimes the inclusions or an inclusion might take up, a great part of the interior of the bacterial cell. So what are these inclusions for? They're generally places where some kind of substance has been concentrated for storage. So lipids and starches, for example, those can be stored as nutrients. So just like we store lipids in our body as sources of nutrition for when a famine might come along, bacteria, some types of bacteria might do that as well with lipids and starches. Hey, when the nutrients are abundant, let's go ahead and take as many in as we can and store up the extra in case we get into a situation where we don't have access to nutrients. Some examples, some bacteria will have inclusions that contain glycogen. Glycogen is a polysaccharide, chains of glucose molecules. You've heard about glycogen before if you've taken AMP1 because our skeletal muscle cells and liver store glycogen as well. Lipid polymers, so you can link lipids together and store those inside these little inclusion bodies that exist in some bacterial cells. And there are some that store other unusual chemical compounds. Interestingly, some photosynthetic bacteria will store gases in here. And if you think about it, if you're a photosynthetic type of bacteria, photo refers to light. You need sunlight as your energy source. And so if you're in the water, you need to be floating up near the surface where you have more access to sunlight. So the gas vesicles in some photosynthetic bacteria appear to act like little flotation devices, believe it or not. All right, so that was just kind of a quick run through on some of the interior components of bacterial cells. And use your study guide to review that information and also what's contained in your textbook there toward the end of Chapter 3. I want to spend a little bit more time, though, on endospores. So what are endospores? So endospores are structures that are made in some bacteria. Okay, not all of them. Only some bacteria make these. And the medically important ones that do so are all gram-positive. So they have that thick peptidoglycan cell wall. And it just so happens that so far, there are some gram-negative bacteria that make endospores, but so far the ones that have been discovered are not medically or clinically significant to us. All right, so what is an endospore? Endo means within. So it's a spore that's made within the cell and often contained within the cell for at least some period of time. One thing, though, is this is not a reproductive structure. This is basically a method for a bacterial cell to go dormant and create a very hardy structure that can withstand harsh environmental conditions. So when you have an endospore, like we see here on this bacillus-shaped bacterial cell, what's in there? The chromosome, so we just talked about the DNA, generally existing as one large circular DNA molecule. And again, a chromosome, the DNA, has all of the genetic blueprints that tell the cell how to function and make all of its parts. So if you're going to go into a dormant state and kind of go into a suspended animation state, you need your DNA. Those are your genetic recipe books. So the chromosome will be in there. And then the rest of it is mainly just a very hardy protective shell that's covering the DNA. So these will form due to stressful conditions. And once that forms, the rest of the cell can die. So the metabolically active portion of the cell can go away, and you're just left over with this dormant endospore with the DNA on the inside. All right, so Endospores tend to form due to stress, harsh conditions, and the bacterial cell can't really actively metabolize and go through its life processes. These endospores can last for very long periods of time, and when conditions become more favorable for the little bacterial cell to regenerate, the endospore more or less germinates and recreates an active cell like you see here. So this is not reproduction because you're going from one cell to one endospore dormant form, and then that can later recreate an active cell again. These things are very resistant to lots of things that we throw at microorganisms to try to kill them, such as heat and harsh chemicals and even radiation. We'll learn more about that when we get to chapter 9 later in the book. So, all right, so we said some bacteria that are medically important make these endospores. They're a huge challenge because they're difficult to control. So if you have a patient who is infected with a type of bacteria that makes endospores, often there are different protocols or procedures that you have to use to decontaminate that patient's room, and you have to take special precautions to avoid spreading those endospores throughout the medical setting where you're working, because unfortunately if another patient gets exposed to these endospores that are really, really hardy, they get exposed to them, those endospores can germinate and cause an infection in another patient. This is a big reason why we need machinery called autoclaves, which are basically like big pressure cookers, steam under pressure being used to sterilize equipment and supplies, waste and so forth in medical settings, because you got to have very high temperatures and harsh conditions a lot of times to kill these endospores. All right, your textbook, this is a diagram from your textbook which is showing the different stages that occur for endospore formation. And I actually don't really care that you learn the detailed steps of endospore formation. I'll just kind of point out some highlights here. So you're starting off here. This is an active cell, so doing all of its metabolism, taking in nutrients, generating waste, and so forth. One chromosome there. So let's say this bacterial cell encounters some harsh conditions. It's depleted of nutrients, for example. The first thing the chromosome is going to duplicate, it's going to replicate, which is just like what would happen if that cell is going to divide. And then the one copy of the DNA, the chromosome, goes to one end of the cell and the other one stays over in the other one. And so far this almost looks like cell division, but the bacterial cell starts to form an immature endospore called a forespore. And then there are additional layers that are added. So there's a spore coat and a cortex. Cortex is like an interior structural layer of the endospore. So you wind up with these, with the DNA in the center and multiple coat layers, endospore coat layers around this. And the chemical properties, the chemical composition of the cortex and the other coat layers makes this spore very, very, very resistant to things that are being encountered out in the environment. And that's really all I care that you know about with regard to endospores. Don't try to memorize the detailed process for how these endospores form. All right, endospores, again, very clinically challenging. So this is an image made with an electron microscope. And so you're looking at one end of a bacillus-shaped bacterial cell, and then here, this darker area here represents the endospore. So you can really see the spore coat there around the endospore. The DNA is going to be in the interior. Here's another picture of an organism called Clostridium difficile, bacillus-shaped organism, and then this thing that looks like a spoon on the end is actually the endospore of that organism that has developed. So there are several pathogens, bacteria that cause disease, that produce endospores, and several of them are in a genus called Clostridium. So this is a genus name I want you guys to be familiar with, and I want you guys to be familiar with these 4 organisms and the diseases that they cause, because they come up pretty frequently. So you got Clostridium botulinum, Clostridium tetani, Clostridium perfringens, and Clostridium difficile. They're all in the same genus, 4 different species. They cause 4 different diseases, which you've heard of Probably all of these, at least 3 of them. I'm sure botulism, type of food poisoning, tetanus, we've all heard of tetanus, commonly known as lockjaw, gangrene, you've no doubt heard about gangrene, somebody getting that after frostbite or some sort of a severe injury. And finally, this one, Clostridium difficile, and sometimes that's pronounced difficile, but it's actually difficile, but almost everybody says difficile, and or commonly known as C. diff. That's how you'll hear it pronounced in a medical setting. This is an organism that is a huge problem in medical facilities and nursing homes. People with weak immunity or people who've been treated with really strong antibiotics are at a risk of developing an infection with C. diff, and it causes a colitis. So anytime you see itis on the end of a word, refers to inflammation. So this is inflammation of the colon, and it's supposed to be very painful, horrible diarrhea, characteristic odor. People, healthcare workers I've had in class who already work with patients who get C. diff, they say, oh yes, you absolutely know from the odor that a patient has it. So that's something you guys are going to run into quite a bit in your medical careers. Then finally, the Bacillus genus of bacteria also has a number of endospore formers, and one of those is Bacillus anthracis, which causes anthrax. And no doubt you've heard of anthrax spores before, because those were being mailed around by terrorists back after the 9/11 attacks. And so that shows you how hardy these spores can be. They were being mixed with white powder and mailed in the mail, and people inhaled them, and those spores germinated in their respiratory tracts and caused inhalation anthrax, which is very commonly fatal. Another member of this genus, Bacillus cereus, is an environmental type of bacteria that can cause food poisoning. So that's another one in that genus. It's an endospore former as well. Endospore former, that's right, not endospore farmer. C. diff, I will, on Blackboard, I'm planning to post a YouTube video, hopefully it's there already, which explains a little bit more about C. diff. And so what happens with this organism, now hopefully by now you've come to appreciate that your body is full of lots of different kinds of bacteria on your skin and along some of your mucous membranes, and especially down in the colon. The colon is a zoo of bacteria. And so let's say you take a really strong antibiotic drug. You've got some sort of bacterial infection going on in the body where it's not supposed to be, and you have to take a really strong antibiotic to get over it. Well, guess what? That antibiotic is also going to kill a lot of those normal friendly bacteria in your colon. If you get exposed to C. diff endospores, and those make their way down into your colon, or maybe you already have them there. C. diff is an organism that already lives at a low population in many people's colons. Well, C. diff, if those endospores are not going to be affected by that antibiotic drug, and so if they germinate after you discontinue taking the drug, C. diff can pretty quickly overpopulate in your colon. So at low numbers, you probably don't even know C. diff is there. When it overpopulates, it can start to cause inflammation of the colon and injure you, and you wind up with this horrible, nasty diarrhea. So it's something you need to be very aware of as a healthcare worker, because there are going to be special precautions that you have to take when patients are infected with this, because if you transfer those endospores, so these patients are shedding these endospores all the time, and if you transfer those to another room, or you bring a patient into that room after the C. diff patient has been discharged, they can very easily become infected with these. Doesn't commonly cause colitis in healthy people. It's usually after you're already sick, you've got weakened immunity. Older people with weakened immunity are more susceptible to it. And if you've been treated with a really strong antibiotic that has allowed this C. diff to get a toehold and overgrow in your colon, then you're more likely to develop that type of an infection. All right, lastly here, some non-membranous organelles. So organelle, now if you've studied cell structure and so forth in a freshman biology, or maybe your AMP1 class, eukaryotic cells like ours, human cells, have lots of organelles, and they're covered with membranes. They're specialized compartments that have particular functions that they do. Bacteria generally don't have membrane-colored organelles. So an organelle is a part of the cell that has a particular job or function, and in bacteria we can refer to those as non-membranous organelles. So ribosomes are an example. Again, all these little spots that you see, those are areas where proteins are built. So you take amino acids and you chain them together to build proteins. Cells need lots and lots of different proteins. We covered that back in chapter 1, and you learn about that in any biology class. Without your proteins, you're going to die. Proteins are the workhorses of a cell. Now later when we get into chapter 8, we'll see that these ribosomes, those little spots you see there, they're machines, but they themselves are actually built from proteins and also a special kind of RNA called ribosomal RNA. And we'll learn more about that when we get into chapter 8. So we talked earlier when we were going over the peptidoglycan cell walls of bacteria that many of the antibiotic or antibacterial drugs that we take when we have a bacterial infection act on the cell wall. And if you can't make your cell wall, you blow up from taking too much water in by osmosis. The ones that don't do that, most of them or many of them, interfere with the ribosomes in bacteria. Our cells have ribosomes too. We've got to build our proteins as well. But lucky for us, the ribosomes of bacteria are different enough from our ribosomes that there are a number of drugs that can screw up their ribosomes without screwing up ours, as long as you take a drug at a proper healthy dose. Cytoskeleton, so again, hard to see probably on the PowerPoint or on the video, but there are little purpley threads shown there on the bacterial cell diagram. Those are fibers, so long, skinny, thread-like proteins. And our cells, human cells, eukaryotic cells, have an extensive cytoskeleton, so you always talk about that when you study eukaryotic cell structure. And it really hasn't been appreciated until fairly recently that bacteria have a cytoskeleton as well. They probably help, they kind of form as the name suggests, a meshwork, a netting type structure inside the cell that helps the cell maintain its shape and structure. So you may wonder, like, why does a cell have a bacillus shape? And it may be that cytoskeleton and the way it's arranged is helping that cell maintain its shape and structure. This is a picture from a previous textbook that we used of some Bacillus subtilis. That's another species in this Bacillus genus, and it's been stained with a special glow-in-the-dark fluorescent dye that attaches to the cytoskeleton. So what you can see there is that cytoskeleton is pretty extensive inside those cells. I do want to make another point. When you see this genus of bacteria, Bacillus, like we just talked about, Bacillus anthracis, yes, that genus of bacteria, they're all related to each other, and that genus name came from the bacillus shape that those bacteria have. But I do want you to appreciate that not all bacillus-shaped bacteria are in that genus. It just so happens that this one particular genus was named for the shape of the cells. There are many, many, many bacillus-shaped bacteria that are in gazillions of different genuses, or genera is the plural of genus. All right, so that concludes this video lecture. The next one, we're going to talk a little bit about the archaea, the other prokaryotes, and just a little bit about prokaryotic classification.